We're delighted to have 4 experts joining us today. Rob Vreers, the CEO at Hubrecht Organoid Technology, Kalpana Barnes, Manager of Cell Imaging Applications at SARTORIUS, David Spector, Professor and Director of Research, Cold Spring Harbor Laboratory, and Martha Shabadze, Group Leader, MRC Laboratory of Molecular Biology. We will begin today's discussion with an introduction from each panelist on themselves, their research, the challenges they face, and their future hopes. Then we will review some of the results from the survey, including key trends in 3D cell culture, 3D cell culture preparation, their applications in drug discovery, and the ethics and future perspectives of 3D cell cultures. So, to begin their introduction, I'll now hand over to Robert Vreers, CEO of Hubrecht Organoid Technology.
Thank you very much, Tristan. Indeed, Robert Vreers, the CEO of the Hub. We at Hub are working with organoids, organoid technology that we developed originally based on adult stem cells. Adult stem cells were identified in the lab of Hans Klevers in the Hubrecht Institute, which is where the Hub originated. The key to the technology and where it is relevant for this 3D and drug development seminar is that the organoids from adult stem cells, epithelial adult stem cells, can be grown without a genetic change. That is a key aspect that we're talking often about 3D cultures, but one of the important features for drug development is that we want to have model systems that are very closely related to the systems that we are using in the clinic and to patients, what we want to develop drugs for. And so we focus the technology that we set up with our organoids on having a genetically stable system and thereby reproducing patient samples. And so just to finish off this very brief introduction, so what we do at Hub is to make biobanks of that resources and use that as a platform for drug development and research into diseases. Thank you, Tristan.
Thank you, Rob. And we will now hear from Kalpana Barnes, Manager of Cell Imaging Applications at SARTORIUS.
So, thank you, Tristan, and hello to everyone, and thank you for this invitation. As highlighted, my name is Kalpana, and I work within the Cell Imaging and Applications Group at SARTORIUS in the UK. I am part of and lead a small team of individuals who are focused on delivering a suite of integrated live cell analysis solutions for those wanting to use 3D or advanced cell culture models in their area of research. And I would say this really involves the development and validation of Incucyte imaging and analytical software, compatible reagents, protocols, and ultimately real-time applications focused at present in the field of oncology and immuno-oncology. These applications go some way to providing biological insight and quantitative measurements of 3D culture cell morphology, health, and function in real time over days and weeks within your tissue culture incubator. And I would say that some of the key challenges faced are around imaging these complex multicellular 3D structures, recapitulating the multifaceted 3D microenvironment through the inclusion of extracellular matrices and additional cell types, automating or industrializing these 3D culture models, and finally quantifying these very dynamic phenotypic observations. And I would say more recently, sort of gaining access to a patient-derived material and the diversity of protocols that are being used to create and maintain such cultures presents a new challenge. So, thank you.
Thank you, Kalpana. And we will now hear from David Spector, Professor and Director of Research, Cold Spring Harbor Laboratory.
Hi, Tristan. Great to be part of this panel discussion. A major focus of my laboratory is trying to decipher the molecular function of long non-coding RNA molecules. these molecules represent a very significant portion of the transcriptome in mouse and in human cells. And we are trying to use organoids as a means to identify critical long non-coding RNAs that may have therapeutic potential in the area of breast cancer. And we are also developing drugs against particular candidates that we are identifying using these organoid models to see if we can perturb the biology of the organoid with the ultimate goal of having impact on patient treatment. Now, of course, the great advantage of this approach is the fact that we can get material directly from patients, both tumor organoids as well as normal organoids from the same patient. And so we can have a direct comparator at the individual level rather than using cell lines that have been growing in culture for tens or 20s of years, which of course we've made great progress using. However, being able to work on human patient samples directly has been an enormous advantage to the research program in my lab, and we hope over the next 5 to 10 years we can see many positive fruits from the initiation of using patient-derived organoids in our research program. Thanks.
Thank you, David. And we will now hear from Marta Shabazzi, Group Leader, MRC Laboratory of Molecular Biology.
Thank you, Tristan, and hello, everyone. So my name is Marta Shabazzi, and I recently opened my lab at the MRC LMB in Cambridge. And my lab is interested in the development of the mammalian embryo, both mouse and human embryos, at the time of implantation. This is really a critical developmental stage when we know many pregnancies fail. However, there are many technical and ethical limitations to study this specific developmental phase. And to overcome these limitations, we are using in the lab 3D cultures of both mouse embryonic stem cells and human embryonic stem cells. And using these models, the question we are trying to address is how epithelial tissue organization modulates pluripotent stem cell identity. And the reason why we want to address this question is because the first task that embryonic cells need to undertake is the formation of an epithelial tissue, as you can see in those images. As soon as the embryo implant, embryonic cells become epithelial. And we really want to understand how those epithelial features, cell-cell addition, cell extracellular matrix addition, polarity, luminal cues, how all those epithelial features are modulating the identity of the pluripotent stem cells and also their ability to differentiate into different lineages. Thank you.
Fantastic. Thank you, Marta. So we will now move on to our first section, Key Trends in 3D Cell Culture. So the first question is, from the results of the survey, the most common disease area studied with 3D cell cultures was cancer. Are you surprised by these results? Kalpana, what do you have to say on this?
I would say no, I'm not really surprised by this survey result. I think we know that 3D in vitro models have been used in cancer research very much, I would say, as an intermediate model between in vitro cancer cell line cultures and in vivo tumours. And I think we're all very acutely aware that cancer drug approval rates are low, almost in single digits. And this is despite significant investment in cancer research and drug discovery and development. So this has really driven the need to more closely align our cellular models used in early lead drug discovery with preclinical animal models and patient tumours. And we know that spherical 3D in vitro models have been described and been used for the last 4 decades. And there is, a lot of literature to support why they're ideal models with replicating key features of human solid tumours with respect to their architecture and the biology, thereby representing, as I say, a more physiologically relevant platform for drug delivery. And as I said, some of these key features point to their architecture with respect to multicellular arrangement and extracellular matrix deposition, which we see in vivo, the closely packed nature of the cells, allowing key cell-to-cell communications and cell-to-ECM interactions. So I think also co-culture cancer models of multicellular tumor spheroids, which incorporate an extracellular matrix and additional cell types such as immune cells, endothelial cells or fibroblasts, go some way to allow researchers to assess functionality of cancer drugs through evaluation of chemoresistance and immune tumor cell interactions. So yeah, I'm not really surprised by that survey result.
Okay, and David, what's your opinion on that question?
I fully agree with what was just said. I'm not surprised at all that cancer is a major area of interest for the organoid approach. As I mentioned already, in the cancer arena, we have been using 2D cultures for a very, very long time. We, of course, have been using mouse models, including PDX models, and spheroids from the early work of Meena Bissell and Senthil Muthuswamy, for example. However, the ability to get samples directly from patients and put them into 3D culture really brings us closer to the area that we are really trying to impact in the cancer field, and that is the patient. And what better means than directly being able to sequester components of the tumor and grow them in a milieu that more efficiently represents the actual tumor that we're trying to study. So it's a very, very important area of focus. And I think a big advantage of using patient-derived organoids is that this whole process is scalable, much more so than mouse models, for example, because we can develop organoids from a very, very large group of patients. We can then put these organoids through drug screens, et cetera, to really, in a more rapid manner, gain important insights at the tumor and patient-specific level. Thank you.
Okay, and another key trend identified was that the most common type of 3D cell culture used was spheroids, and a large proportion of respondents used both spheroids and organoids. Why do you think this is? Let's start this with Rob.
Yeah, I think I was a little surprised by it, to be honest. I think that it might be a chronological, thinking of the questions answered before. Spheroids have been around for a bit, of course, about 10 years, maybe 20 longer than organoids, if we count the very first. And therefore, maybe that's not very surprising then that it is used more still. What I think is maybe important about this is that because they have one has been around for a bit longer, and also the definitions have been a bit maybe a bit confusing, especially in the beginning. Spheroids are aggregates of cells and often still cell lines that are put together and form the first 3D systems, and that indeed gave us different answers than before, because now we could look at cells that were interacting differently, at least with each other than they were when we were using 2D plastic culture systems. However, organoid was, as far at least as we're concerned, and a big step from that, simply because now you have a culture system that allows this genetically stable patient-derived system to grow in a morphological system, morphological composition that is very similar to normal epithelium in our case, and neurons or whatever people are using in other organoid systems. And therefore they are really not the same anymore as spheroids. That maybe explains why people are using both. Whereas the spheroids are relatively close to the traditional cell line models, or organoids really are a transition to in vivo models to patient-like models. And I think, and that maybe as a last part in connection also to what was said in answer to the previous point, the organoids as opposed to spheroids, and then looking at the disease models that are studied, cancer, of course, is something that we could traditionally grow better in culture because cancer cell lines were one of the few things that we could actually make. The big difference to an organoid is that, and this maybe explains why there's so much people using cancer still. However, I think, and this is the importance of seminars like this, it's important to realize that because of the organoids, we can now not only grow cancer cells, but we can actually grow healthy cells as well. And that allows us to grow all the other disease models as well, and hopefully result in also all the other fields, in infectious diseases, I guess very relevant now, but also IBD, COPD, genetic diseases, can use these organoid models because they allow a genetically stable, healthy cells to be cultures in addition to the traditional cancers that we were already growing. Thanks, Tristan.
Okay, thank you, Rob. And Marta, what is your opinion on the use of organoids and spheroids?
Yeah, I have to say I wasn't really surprised to see this. I mean, I think it's important to consider what type of question you want to address, and depending on the question, you may want to use spheroids or organoids. As it has been said already, spheroids have been there for quite some time already. They are relatively simple to use, and they really allow the molecular characterization of the mechanisms of polarization, luminogenesis, all the fundamental work of people like Keith Mostov. So for those labs that are really interested in the biology of a cell and the biology of epithelial tissues, spheroids may be just a great tool, and they don't really need to go to the organoid. Whereas those labs that are more interested in clinical applications or disease modeling may prefer to use organoids. So I think both models have advantages and disadvantages, and the great thing would be able to combine them and use them depending on the specific question, scientific question.
Thank you, Marta. And David, what was your opinion on this question?
I think, you know, as Rob said initially, spheroids have been around for a much longer period of time, and so I think that really is why you know, people have been using them more. However, over the last number of years, I think that the number of labs that are beginning to use organoids is increasing, and I think this will just continue to increase because, you know, they are a fantastic system. And just getting back to the issue of cancer and the use of organoids, I think the impact of organoids in the field has already been recognized in some ways by funding agencies among multiple countries. And what I'm referring to here is the Human Cancer Models Initiative, which is an international consortium that has been set up by multiple funding agencies, the National Cancer Institute in the United States, CRUK in the UK, and of course the Hub in the Netherlands, and the Wellcome Sanger Institute. And together, these funding agencies are providing support to a group of model development centers, including the Hub, CRUK, and Wellcome Sanger Institute, Cold Spring Harbor Laboratory, and the Broad Institute. And the goal here is to develop models which can then be deposited at the American Type Culture Collection for distribution to make these models readily available to the entire scientific community. And I think this is already in place, and many models are out there. And I think, you know, the availability of these models for laboratories around the world is going to have a dramatic impact upon the use of this system and also what we can learn from this system. So I think the future looks extremely bright for organoids.
Fantastic. Thank you, David. And Kalpana, do you have any words to add on this subject?
I think most of the comments have been captured by the panelists thus far. I think perhaps one thing to highlight is that I think we all understand why spheroids have been so impactful to date, but I think organoids are very much catching up, and I think that's really down to the advancements and understanding of how to culture them, how to cryopreserve them, and we are seeing their use and application in disease modelling, infectious disease, personalised medicine of late. But I think something also to be mindful of is that human diseases that are difficult to model, for example, in animals, could potentially be studied using patient-derived organoids. So I think that is something very powerful going forward.
Fantastic. Well, we'll now move on to our next section, so 3D culture preparation challenges and best practice. So the majority of respondents produced their own cultures in their labs and used cultures built mostly on scaffolds. David, do you produce your own cultures? And if so, why do you not purchase commercially available cultures?
We do use our own cultures. The main reason is that we want to obtain material directly from patients from which we can have background information in terms of mutations, et cetera, as well as pathology. And so, for us, those models have not been currently available. As I mentioned, now that a number of centers are developing models which will be available to the scientific community, I think in the future, many will choose to purchase these available models. But at the moment, our goal was to develop models directly from patients and being able to obtain both normal and tumor from the same patient.
Thank you, David. And do you agree with the more prevalent use of scaffolds? What are the additional benefits or disadvantages to scaffolds, and what do they bring to the production of 3D cell cultures? Kalpana, would you like to share your thoughts on that?
Yeah, sure. I think one important contribution for attaining this closer to the in vivo behavior of cells when grown in 3D comes very much from the matrices and scaffolds used. They provide structural, physical and biochemical support and an environment allowing cells to proliferate, differentiate and promote these key cell-to-cell and cell matrix interactions. There are several scaffolds available, both biological and synthetic, and the choice is very much dependent on the cell type and the nature of the study. So biological scaffolds use naturally derived extracellular matrices, such as matrigel and collagen, to promote cell attachment and organization of cells in 3D cultures, whereas synthetic scaffolds use synthetic hydrogels or other biologically compatible material to form a physical support for the 3D culture. So there are some key advantages. As I said, they help to better recapitulate the in vivo microenvironment, and there are some really good commercially available sources. Compared to synthetic scaffolds, biological scaffolds like matrigel, for example, can provide this physiologically relevant microenvironment rich in extracellular matrix proteins such as laminin, collagen, protoglycans, and soluble growth factors. And matrigel, I think we all acknowledge, is widely used and almost considered a gold standard scaffold for use in 3D cultures. However, it does come with some disadvantages, and I'm sure we're all very mindful of these. It's a natural matrix. It's poorly defined with respect to these growth factors and endogenous components. There's considerations around batch-to-batch variability during manufacturing, the complexity in the composition, which is often ill-defined, and that makes it difficult to define which signals are promoting cell function. gelling temperature is a consideration, and I think especially when automating these cultures, this becomes a key challenge. The opaque nature of the gel can often present a challenge and difficulties with imaging cultures that use these gels. And of course, Matrigel is animal-derived, so ethics is a concern. And I would say that it really limits the use of cells grown in it for clinical applications such as regenerative medicine. We said it's highly heterogeneous in composition. Therefore, the precise modulation of the physical and biochemical attributes it offers is not possible. I think all of these factors provide a real strong rationale to develop a defined basement membrane extract substitute. I think synthetic scaffolds can address or circumvent some of these disadvantages which the biological scaffolds present, in particular Matrigel. They can minimize some of the poor reproducibility between batches I spoke of. They can allow some fine-tuning of the biochemical and physical mechanical properties, which you can optimize for your 3D culture. And as I understand it, they provide a high water content, which goes some way to facilitate transport of oxygen, waste, and nutrients in the culture itself. And of course, an obvious disadvantage is that they don't have these endogenous factors present in Matrigel I spoke of. So they often serve mainly as a template to regulate cell behavior.
Fantastic. Well, thank you, Kalpana, for that really in-depth look at the at Matrigels. Marta, do you have any words to add about Matrigels, their popular selection, and their advantages or disadvantages?
Yeah, so as Kalpana said, Matrigel is really a gold standard in the field, I would say. Matrigel was already discovered in 1977, so it is not surprising that throughout the years people have been using it. It is very easy to use. You can just buy it and use it, and it works really great. But as Kalpana said, there are many limitations. For example, in my field, it has been shown that in Matrigel, cells are more heterogeneous. So if you want to have a homogeneous culture of stem cells, it's better to use hydrogels and more defined gels. So that's another limitation to add. And I think we will see in the future that the trend will change, and more labs will start adopting hydrogel technologies instead of Matrigel, because you can control them much better, you can manipulate them much better, and you can really dissect the different contributions of the different parameters of the gel into the behavior of the cells. So for the time being, I'm not surprised looking at these results, but I think things may change in the future.
Okay, thank you, Marta. And the hanging drop method was the most prevalent of all the scaffold-free techniques selected. Why might this be, Kalpana?
I think Marta on the panelist has a far more in-depth insight to that, so I may well ask Marta to respond there.
Yeah, so again, just talking about development, the hanging drop method has been used for a really, really long time. It was used to create embryo bodies originally. These are just collections of embryonic stem cells that are allowed to differentiate. They just form these aggregates, and then using those aggregates as a starting point, we can generate different types of differentiated cells. So this method has been around for quite some time and is really, really powerful. And even nowadays, some of the organoids that people use, like brain organoids, they start with this embryo body step. So yeah, it's a really critical method, the hanging drop method, to generate embryo bodies for many different applications.
Fantastic. Thank you, Marta. And on to our next section, so applications in drug discovery. The top 4 advantages of using 3D cultures in drug discovery were, first, the avoidance of animal model ethical issues, second, the cost versus animal model studies, and joint third, the ability to control concentration gradients in the culture, extracellular matrix and the ability to conduct rapid drug screening. So first off, I'll start this one to Rob. Do you agree with this, and what do you think is the most important benefit of 3D cell cultures?
Well, I can definitely see the first. So the animal models, organoids are intended to replace animal models. I think on the long run, we think that they're human, and therefore we are finally done with all the artifacts that we get when we generate data from animal models. And of course, the ethical concern that we have with animal models, and I think if we have a way of getting rid of those, then that would be great. In our lab, in our academic lab, Hans Klever's lab, where we originally developed the organoids, you see this trend that was, of course, a mouse genetic labs for many years since the start of organoids. They have, in a large part, gone, and it's all organoid models derived from the patients now. I think the one thing that is missing there, of course, a lot of advantages to organoids over traditional models. I would see the key point, and that is actually missing from the list, is that it's a patient representative model. So whereas in the older models or even in animal models, if you want to know whether your data is relevant, if your findings are representative of what happens in the clinic, you cannot do that. But since organoids are directly derived from patients, simply by, and David already alluded to that, you make a model system, you go back to the clinical records and see what happens to the patient in the clinic in response to drug, and you can see whether your organoid model in the lab does the same thing. And since we and others have shown that this is indeed the case for cancer, and cystic fibrosis was the disease that we started this on, I think that is the main point in drug discovery for using an organoid, that now finally in the lab you can give much more value to the data that you're generating, simply because you know that you can validate this model directly to the clinical responses that you see in patients. So although I think that an advantage is that we can indeed finally get rid of using animals, I would say the key point of using organoids in drug development is that we can greatly speed up, hopefully, drug development by increasing the success rate of developing drugs preclinically that will actually work in the clinic.
Thank you, Rob. I'll now hand it over to David, who can obviously speak a little bit more about the patient-derived benefits of it.
Yes, I totally agree with everything Rob said. I mean, animal models, of course, have provided us with great insights over the years, not all of which, of course, have been translatable to the human scenario. However, the use of these patient-derived organoids is extremely valuable. They're much more scalable than using mouse models. We can look at a huge cadre of patient samples from which we've developed organoids from, to assess various drugs that are, standard of care, or new drugs that are being developed, combinations of drugs, etc. We can use high throughput screening methods for this to look at multiple drug concentrations. We can do high throughput imaging with these organoids over time, etc. So our end take home from using these models is going to be much more rapid than were we to use mouse models. In some cases, we wouldn't even be able to use that large number of mouse models. It's much more cost effective. And in addition, it's going to directly relate to the patient more so than what we derive from using animal models. So I think we're going to see a great advance in the ability to translate our scientific discoveries from the laboratory to the patient using these organoid models in a variety of different diseases. Thanks.
Thank you, David. And Marta, do you have anything to add on this question?
I mean, I could comment on the applications of 3D cultures for development. And I would say that here the key advantage is that we can actually follow a bottom-up approach when we are doing research. So if we think about an embryo, there are many different types of cells. There are chemical interactions going on, physical interactions going on. So it is really difficult to dissect the complexity of development using the embryo. However, if we use 3D cultures, we can actually tease apart all those influences and follow up a bottom-up approach in which we can progressively increase the complexity to really know what is the contribution of specific variables to a given phenotype.
Okay, fantastic. And Kalpana, do you have any last words to add on that?
I mean, I think the rest of the panelists have covered the response in some depth. I think just a couple of points on the survey with regard to avoiding animal model ethical issues and cost versus animal model studies. I think we all know that everyone within drug discovery and biomedical research tried to adopt that approach by application of the 3 R's, so replacement, reduction, refinement. And we all know that public opinion polls routinely show that support for animal research is conditional on the 3 R's being implemented. So with regard to replacement, I think methodologies such as 3D culture models and technologies which avoid or ultimately replace the use of animals in experiments is a really good fit. We know animal studies are not only lengthy and costly, as we've just heard, so the introducing of 3D culture models and their use arguably with more relevant cell types, as we've just heard, whether they be stem cells, primary cells, or patient materials, much earlier in the drug discovery continuum can mitigate these challenges and, importantly, obviate the animal versus human species differences.
Fantastic, thank you. And what techniques in the drug discovery process do you think 3D cultures lend themselves to best, Kalpana?
So with regard to processes, I think my interpretation of the word processes sits along the sort of drug discovery pipeline. That's how we've interpreted this. So I think 3D cultures is showing potential and promise in early phase drug discovery and is already being used in areas such as target identification, which is often considered a rate-limiting phase in preclinical discovery. So here, 3D cultures can be used in the identification of novel drug targets, whether they be proteins or pathways, via omic panning. And compared to 2D cultures, the gene expression patterns in 3D cultures are one step closer or align better with in vivo expression profiles. So they also have a place and add value in the phase of compound screening and triage for hit identification. I think here, through the adoption of phenotypic screens using more relevant cell-based models, provides a means of identifying new first-in-class drugs. And in the area of lead selection, efficacy and safety assessment, I think this phase really relies on cost-effective models which reliably predict the efficacy, toxicity, and pharmacokinetics of compounds in humans. So here, I think 3D models have an important role to play through reducing the number of animal tests required ahead of preclinical studies. And personalized medicine, I think, is another area where cells or tissue biopsies used in vitro 3D cultures can be used to identify drugs showing patient-specific efficacy. Thank you.
Thank you, Kalpana. And moving across to the challenges of 3D cultures in drug discovery, the top 3 challenges of using 3D cultures were identified as reproducibility, labor intensity, and the visualization of cultures with automated imaging systems for high content screening. Do you agree with this, and what do you think is the biggest challenge of using 3D cultures for drug discovery? Rob, we'll start with you.
I think that indeed, well, especially the first one, when we're using organoids, and main point is that they are a little bit more complex to grow than a traditional HeLa cell or a 2D system. So that means that the reproducibility is determined very much by the reagents that are used, by adherence to protocols, etc., in order to be able to get reproducible results. And so also from our side, so when we initially used organoids, we had to develop the tools and the reagents to make sure that that was actually possible. Initially, it looked actually quite difficult to get the patient-derived sample and then to be able to grow it for long term and maintain this reproducibility. But indeed, what we found is that it is not so difficult as we might have thought. It is simply using the protocols very strictly, using reagents that are well described, that people have used or that are tested, and then you can have a very reproducible result. In the collaboration with the NCI, Cold Spring Harbor, CRUK, and Sanger Institute that was already mentioned is a very important part for us also to be able to sort of make sure that all the tools are there, the organoids are there, and that everyone actually in their own lab is able to use the system and deal with the challenges of that reproducibility. I think that for me is the challenge that's still there. That is, I think we have the reagents now and we have the protocols to do it. Nevertheless, developing these assays now associated with it, transferring protocols, exchanging protocols between labs continues to be a key aspect. And I think that there is one thing that, because the technical part of it, of course, brings you to a certain extent to where you want to be in order to deal with these challenges. Nevertheless, another key point, and that is maybe a positive one, is that now we have these very patient-like models that, as said in the previous one, we can actually see that it can be used for personalized medicine. So we use it in the Netherlands to identify patient responses to cystic fibrosis drugs, and they can subsequently be treated, and indeed that works very well. But it also shows how different patients are, how different samples are, and how sensitive samples are, maybe also in real life, to different changes, to exposures, to different media, etc. So although on the one hand, of course, it is a challenge to make them reproducible and to deal with these heterogeneous samples, I think there's also some questions on it. It in itself is actually it is an indication of this more in vivo-like system that has this very strong heterogeneity in cell types, and when we're talking about cancer, in genetics, and that is now represented in the cultures as well. And then, and I think it directly relates also to the imaging and the labor intensity, the other challenges that were mentioned. We have, of course, in the labs here. It is more labor intensive because it requires quite a lot of QA to make sure that you have the right sample and that you keep testing it, that it is still the right sample. When we do imaging, there are so many features. It is now possible to do high throughput screening and imaging, but it does mean that we generate an enormous amount of data that we then have to analyze and understand. So I think these are, as far as I'm concerned, challenges, but also are in fact why we wanted a patient-like model. We wanted to have a healthy system, and then now last bit, going to safety. What we find in a European project, a Human Cell Atlas project, which we started to generate 100, for instance, healthy intestinal models, when we do our tox studies, and that we know that from patients, of course, but now also in organoids, we see that 10 healthy organoids are 10 different patients and therefore give 10 different results. So we actually have to learn to deal with that. There is not so much as a golden standard as we used to use for hepatocytes anymore, but we actually are dealing with a patient-like population, and we have to adapt our lab techniques accordingly. Thanks.
Okay, thank you, Rob. And Kalpana, do you have any insights into how to overcome these challenges?
Yeah, I think we've just heard around implementation of more relevant cellular models in early phase drug discovery will certainly improve the quality of molecules entering preclinical phase of the drug discovery pipeline. And we've already discussed why and how 3D models can address this. So factors which hinder or preclude adoption include reproducibility, cost, time to set up, low throughput capacity. So being mindful of these factors and addressing them during model design and optimization is key and hugely valuable. There is also, I would say, limited detailed characterization and cross-comparison of complex models to illustrate added value relative to more simple 3D models. So why adopt them if my 2D assay is less labor-intensive, is cheaper, is a question you may well be faced with. So I think picking up on some of the comments we've heard earlier, I think methodical and potentially collaborative studies on the characterization and standardization of 3D culture systems will be of value. A couple of examples to note, we saw this with the MIP-DILI consortium, which ran across 2012 to 2017. And here, academia and pharma pulled together and collaborated on improving the models used for liver toxicity testing, for example. And we've heard already how the Human Cancer Models Initiative Consortium are building and sharing models, protocols and knowledge to advance and refine 3D model development. So these are all positives and could address some of these challenges. And I think while target-based drug discovery is still a valid approach, I think going back, the adoption to phenotypic screening provides an alternative, unbiased option. And I think your choice of technology or instrumentation used to capture and quantify these phenotypic responses is important. You may well be using, as we've heard, very high value, precious, patient-derived cells, which could be limited in supply. So the data you capture from these cell types needs to be of high value, therefore ensuring high quality downstream analysis. And how you capture and interpret the multi-parametric data can be a challenge in itself. So data acquisition and automated data analysis shouldn't really be a bottleneck for you to extract meaningful pharmacological data from your experiment. So we know high content images often require experienced super users. They are costly, both of which can preclude adoption in drug discovery. And I think if you're using an image-based approach, I think there are some advantages to using the same instrument or system during the optimization of your 3D culture, any manipulations that you do, and right through to the assay phase of your workflow. So having that continuity with instruments being used is hugely beneficial. And I think finally, how you interpret this multi-parametric data, as I said, can be challenging. And I think if you think about using multivariate analysis approaches, these could allow you to reduce the complexity of the data to a simple consumable into one or two-dimensional space, thereby returning more interpretable values for your study. So I think all of this ultimately will help you achieve or lead you to generating faster decisions from your culture.
Fantastic. Thank you, Kalpana. So now on to our final section, ethics and future perspectives. So do you agree, Marta, with our survey respondents that the transplantation of human-derived 3D cell cultures into animals is the biggest ethical issue in the field of 3D cultures?
Well, I think that in the context of development, the use of 3D cultures the biggest ethical issue is the generation of stem cell models of the human embryo. So we can actually in the lab study human embryos until day 14 only. So this means from fertilization all the way up to gastrulation when the body plan is established. But now what happens if we take stem cells and we create a 3D model that has both the embryonic compartment, the extraembryonic compartment, and what if that model recapitulates the process of gastrulation and beyond? Should the day 14 rule apply or not? And if not, what would be the limit to that kind of experiments? Should there be a limit? I think we still don't have the answers to these questions, probably because this research is happening now, is something really recent. But I think as a community we will need to find answers. So, I mean, the audience didn't put this as one of the biggest ethical issues, but I think it is definitely one of the biggest ethical issues that we should consider.
Okay, thank you for that, Marta. And what do you think is the most needed improvement in 3D cultures, David?
I think right now a critical aspect is standardization. Many labs are using different media or matrices, and obviously this is going to impact the gene expression profile of the various organoids that people are growing. And, you know, while this is fine as long as they, you know, as a given state what conditions they're using for their organoids, I think what the field really needs to come to a more standardized, you know, medium. These media are quite complex, and we need to come to, you know, one or a very few matrices so that we can more easily compare results among laboratories, and also, you know, the preparation of the media is critical, as it is a very complex medium. For example, in my lab, I have one person that, you know, prepares all of the media for everyone who's growing organoids, just to be consistent, so that there are not any individual to individual differences in the way things are being made up. So I see standardization as a really critical component of this work going forward. Thanks.
Fantastic. Thank you, David. Rob, where do you see the field of 3D cell cultures, particularly their use of drug discovery, in the next 5 years?
Thanks. Yeah, I think that well, in this hour we've covered quite a lot of things that we can do, and will probably improve drug discovery in the next 5 years. But there's one thing that for me is particularly interesting, and we did not touch upon very much yet, and that is that because we now have these patient-derived models that seem to be very representative of what happens clinically, we can actually preclinically do part of the work that we would normally do in clinical trials. And so in the lab we call that a preclinical clinical trial, and it basically means we have, for instance, a biobank of 150 colon cancer patients that has this representation toward the actual population that is treated clinically. And what will help us to improve drug development and speed up the preclinical process, or improve the clinical success rate, is when we take our drug candidates and test them on this library, on this reservoir, on this resource of different patients, and then see what will be a clinical result later in order to improve that. And so this basically improvement of the clinical phase, or shortening of a clinical phase, by being able to add this into a much more scalable system. David already mentioned it. It allows you to do combination therapies. It allows you to test drugs of different companies even, actually. Will, I think, be the biggest influence on drug discovery in the coming 5 years, when we actually start using that full potential of organoids by looking at it as a clinical population. Thanks.